Hello students, hope you're liking videos on my YouTube channel. Please do write a comment on what changes do you expect in my upcoming videos. In this presentation, we are going to discuss chapter 6 of class 10 science, life processes. This is part 1 of my presentation on life processes. We'll be discussing here what are life processes, why life processes are integral part of life, and nutrition in plants, amoeba, and human beings. Students, let us see some of the processes which are occurring in non-living organisms and similar to living organisms. Like you have seen a car moving. If it is moving, still we call it non-living. You have seen a building is made to grow by a mason, but still we call it non-living because all these processes are not occurring on their own. They are done by human beings or they are made to grow, made to move and so on. What about living organisms? You have seen a living organism like a dog is moving. If it is moving, it is living organism. But what about if it is sleeping? Still we call it living? You may probably say if it is breathing, we call it living. But have you seen a plant breathing? Probably nobody of us have. So if we haven't seen all these activities, why we call them living? In our earlier classes, we have seen that all living organisms are made up of cell, tissue, organ system, and so on. What happens if this system is broken? Like if somebody happens to meet an injury, this injury is repaired by on its own. So the maintenance of the cells or tissues is also done by living organism. That means there are specific processes which are occurring in living organisms. Let us learn about these specific processes which maintain the life of an organism or the life processes of living organisms. What are life processes? The processes that are essential for living and maintenance of life in an organism are called life processes. These processes include nutrition, respiration, circulation or transportation, excretion, control and coordination, reproduction, response to stimuli by nervous system. All these we'll be studying one by one in my videos. Let us study the first process, that is nutrition. Nutrition is not just getting food. It is the process of obtaining food, digesting it, absorbing nutrients from it, and then throwing the waste. All these processes are part of nutrition. Nutrition is done by two ways in organisms. The first way is some organisms use simple food material from inorganic sources to produce their food. Plants and some bacteria are examples of this type of nutrition. This type of nutrition is called autotrophic mode of nutrition. Plants do it by photosynthesis. The other mode is in which organisms utilize the complex substances which are produced by plants. They utilize it after breaking it. Examples are human beings, amoeba, and many other organisms which are not autotrophs. This type of nutrition is called heterotrophic mode of nutrition. Let us see what is autotrophic nutrition. Autotrophic nutrition is a process by which plants prepare their food. They prepare their food from carbon dioxide and water in presence of sunlight and chlorophyll. The plants obtain carbon dioxide from the atmosphere through stomata. The water and mineral molecules they obtain from the soil through the roots. Chlorophyll is present as green pigment in the leaves. The reaction takes place in presence of sunlight. During the reaction, the water molecules split into hydrogen and oxygen. This hydrogen combines with carbon dioxide to form carbohydrate, which is C6H12O6, or the food which is formed in the leaves. The oxygen, which is formed by splitting of water molecules, is released into the atmosphere through stomata. The excess water, which is left in the leaves, is released through stomata by a process called transpiration. This reaction you must learn for your exams. One more question that is asked is the process of photosynthesis has these three events. So what are the three events? These are mentioned in your textbook. The first point is absorption of light energy by chlorophyll, conversion of light energy to chemical energy, that is formation of carbohydrate molecule, and splitting of water molecules into hydrogen and oxygen. And the last point is reduction of carbon dioxide to carbohydrates. Before proceeding to study other life processes, let us study some of the important terms that are used in figures in biology. Here I have taken a lady's finger. If it is cut lengthwise or along the long axis of the finger, the section is called longitudinal. And the appearance of the section will be like this. But if it is cut perpendicular to the length, the section is called cross section or transverse section. We will get circular rings of lady's finger in this transverse section. On the other side of the page, I have taken an image of the front view and the back or the rear view of a leaf. You will be anxious to know why I have taken this leaf. Though the appearance of the leaf from the front side and the back side is slightly different, there are remarkable differences in the surfaces if we cut the transverse section of the leaf. The transverse section will give you an upper surface, a lower surface, and in between these two surfaces, tissues are embedded. So let us study in detail the transverse section of a leaf. The upper surface of the leaf contains an upper epidermis, which is protected by a cuticle. The lower surface of the leaf also has a lower epidermis, which is also protected by cuticle. In between, there are tissues. The upper tissue is palisade tissue, which has oblong cells. On the lower side, the cells are circular. These are called spongy tissue. In both these tissues, large number of chloroplasts are filled. Chloroplasts give green color to the leaf and are used in photosynthesis. In the center of the section, you can see a vascular bundle. Vascular bundle is present in the veins of the leaves. This vascular bundle contains xylem tissue, which transports water in the plants. It also contains phloem, which transports food to various parts of the plant. On the lower side of the leaf, you can see an opening. This opening is called stomata. On the upper surface, stomatas are few in number. Here I have not drawn even a single stomata, but they are few on the upper epidermis and more on the lower surface. This opening of stomata is guarded by guard cells. That means the opening and closing of stomata is controlled by the guard cells. In science, every fact is supported by proofs. I have told you that in plants, photosynthesis is done in presence of chlorophyll. Let us get a proof for this. We'll take a potted plant with variegated leaves. Variegated means the leaf with colored spots. We'll keep the plant in dark for 2 to 3 days. Keeping the plant in dark de-starches the plant. That means the starch which was produced by the plants is utilized by it and the plant is left with no more starch. Now we'll keep the plant in sunlight for photosynthesis. After 6 to 8 hours, we'll plug a variegated leaf, mark its green portion with a marker. We'll boil the leaf in water to make it soft. We'll again boil it in alcohol. Now here we have to take a precaution. Alcohol is inflammable. So we have to keep the alcohol beaker in water bath. This process will decolorize the leaf. The leaf will turn white. Now we'll wash the leaf in water and dip it in iodine solution. After it is dipped in iodine solution, we see that the green portion of the leaf turns blue-black, while the rest of the portion which was colored do not turn blue-black with iodine. Iodine gives blue-black color with starch. Here you can see the variegated leaf with the green portion. The rest of the portion is yellow with iodine. With iodine, it is changed to blue-black. The portion which was having other colors did not turn blue-black with iodine. This confirms that chlorophyll is necessary for photosynthesis. Let us do one more activity to show that carbon dioxide is taken by plants for doing photosynthesis. For this, we'll take 2 plants of same size and variety. Keep the plant in dark for 2 to 3 days. Keeping the plant in dark will destarch the plants. After destarching, place a watch glass containing KOH by the side of one plant and cover the plants with bell jar. KOH will absorb the carbon dioxide inside the bell jar. Use Vaseline to seal the bottoms of the jars so that no more air can enter the bell jars. Keep the plant in sunlight for photosynthesis. After 6 to 8 hours, plug a leaf from each plant and repeat the iodine test. Iodine test is done to check the starch in plants. First, we'll boil the leaf in water. This will make the leaf soft. Then boil it in alcohol, keeping it in water bath. This will decolorize the leaf, and then we'll put few drops of iodine after washing with water. We are surprised to see that the plant where KOH or potassium hydroxide was kept did not show the blue-black color with iodine, whereas where no KOH was kept, the leaf turned blue-black with iodine. This experiment confirms that carbon dioxide is necessary for photosynthesis. Let us learn another mode of nutrition. It is heterotrophic nutrition, in which organisms obtain food from plants or other organic resources. In heterotrophs, chlorophyll is absent as they do not have to prepare food. Heterotrophic nutrition can be classified into various types. The ones which are given in your textbook are: first one is parasitic nutrition. In this, organisms obtain food from another living organism, and by obtaining food, it harms its host. Examples are tapeworm, leech, and cuscuta. You know, when leech absorbs blood from its host, it harms the host. Cuscuta is a climber which climbs on plants, and it harms its host plant. Similarly, another mode of nutrition is saprophytic nutrition. In this nutrition, organisms obtain food from dead and decaying organisms. It can be dead plants, it can be dead animals. Many species of bacteria and fungi get the food by saprophytic nutrition. The third mode of nutrition is holozoic nutrition. In this nutrition, the organism obtains solid food. Amoeba, frog, insects are all holozoics. We are also holozoics. Let us study the heterotrophic mode of nutrition in amoeba. Amoeba, as we know, is unicellular. Unicellular means it has a single cell body. Though it is single celled, it is doing all the activities which a multicellular organism does. When the organism senses its food, it sends extensions of finger-like projections around the food. And these finger-like projections are known as false feet or pseudopodia. The false feet or pseudopodia surrounds the food, engulfs the food, and forms a food vacuole inside the cytoplasm. This process is known as endocytosis. Amoeba is doing holozoic nutrition because it is getting a solid food. The food is inside the food vacuole is digested by secretion of digestive enzymes. It is converted into small particles. After it is digested, the food particles are absorbed by the cytoplasm through diffusion, and energy is obtained by amoeba from the nutrition. The waste which is left in the food vacuole comes to the surface of the cell and is ejected out. The process is known as egestion. Let us study heterotrophic nutrition in human beings. Nutrition in human beings consists of 5 processes: ingestion, digestion, absorption, assimilation, and egestion. Ingestion is a process in which the food is taken in. So the process starts from our mouth. When the food is kept in the mouth, the food is cut and chewed by teeth. That means it is broken into smaller pieces. Do you know how many teeth we have? We have 2 incisors, 1 canine, 2 premolars, and 3 molars on one side of the jaw. That makes a total of 8. A total of 16 teeth in one jaw and 16 in the other jaw. So we have a total of 32 teeth in adults. The tongue helps in tasting the food, and it also helps in mixing the food with saliva. Saliva is a secretion which is secreted by salivary glands. You can see in this figure, salivary glands are shown by yellow color. So saliva is secreted from these glands, mixes with the food, makes it soft, and it contains an enzyme known as salivary amylase, which converts starch into maltose. So digestion of food starts in the mouth itself. That is why when we keep the food chewing for long, you feel a sweeter taste. The food, which is soft now, is in the form of a ball and is sent inside the esophagus by the muscular tongue. Having noticed that esophagus is continuous with stomach, small intestine, and large intestine. So it's the same continuous tube which is increased diameter at some regions. Like in stomach, the diameter is remarkably more. The esophagus consists of muscles. These muscles contract and relax to push the food forward. This movement is called peristaltic movement. Peristaltic movement is followed in the whole gut. The food is pushed from the esophagus into the stomach. The movement of food is controlled by a pyloric sphincter present in between the juncture of esophagus and stomach. Stomach secretes 3 juices: mucus, hydrochloric acid, and pepsin. Mucus protects the lining of the stomach from hydrochloric acid. Then why hydrochloric acid is needed in the stomach? It is because it kills many bacteria present in the food and it makes the food acidic so that pepsin can act on proteins. Pepsin breaks the protein molecules into smaller protein molecules. Then the food is pushed in batches from the stomach into the small intestine. Small intestine has three parts: duodenum, jejunum, and ileum. The movement of food from the stomach into the small intestine is also controlled by a sphincter present at the juncture of stomach and small intestine. The first part of the small intestine, duodenum, receives bile juice from the liver. Bile juice makes the food alkaline and it breaks big fat molecules into smaller fat molecules. This is known as emulsification of fat. Pancreas secrete pancreatic juice and it is also poured in the small intestine at the duodenum region. This juice contains trypsin and lipase. Trypsin act on proteins to break them into amino acids, but trypsin can act only in alkaline medium. So this was done by bile juice in this region. Lipase breaks the bigger fat molecules into fatty acids and glycerol. Walls of the intestine has glands which secrete intestinal juice. The enzymes present in the intestinal juice completes the digestion by converting proteins to amino acids, carbohydrates to glucose, and fat to fatty acids and glycerol. Intestine is a long tube. It is a long coiled tube present in the abdominal cavity. Herbivores have longer intestines than carnivores. It is because they have to digest cellulose. When the food reaches the ileum, the last part of the small intestine, the food is completely digested. The walls of the ileum have many folds. These are known as villi. Villi increase the surface area for absorption of digested food. You can see one cell of villi contain so many microvilli to increase the absorptive surface of the villi. From the villi, the food is absorbed and it is passed on through the blood vessels present in the villi to each cell, from where the body gets energy. The unabsorbed food is passed on to the large intestine, where water is absorbed from the undigested food. The unabsorbed, undigested food is removed from the body through an opening called anus. The opening of anus is controlled by anal sphincter. I hope all of you have understood the chapter Life Processes till Nutrition, Nutrition in Plants and Nutrition in Human Beings. If you have any doubts or queries, please write in the comment section below. Like and subscribe my channel to get the notification of my forthcoming videos. Thank you.